So, what happens once we form that pyruvic acid? It's dependent upon the state of the cell. Okay. And by that I mean it relates to if there's oxygen or not. Okay. If oxygen is scarce, and this can happen, we will go through what's called anaerobic fermentation, okay, where we take pyruvic acid and we reduce it to form lactic acid. Okay. And as you know, oxidation, which we just said, meant losing electrons. Therefore, reducing means gaining electrons. Okay. That's not the preferred mechanism, but we're going to talk about that on the very next slide. What we prefer is to go through the aerobic pathway, which means that oxygen was plentiful in the cells, and there we take the pyruvic acids that we made in glycolysis and transport them into the mitochondria. Okay. We then go through the formation of acetyl-CoA. We call this the transition reaction. Okay. That then brings us to the Krebs cycle, and we'll talk about the Krebs cycle after we talk about the formation of acetyl-CoA. That's what we call it, the transition reaction. But first, let's talk about the simpler pathway, which is the anaerobic pathway. Now, anaerobic conditions can happen. Example is muscle cells during strenuous exercise. We're talking pretty darn vigorous exercise here. And what ends up happening is we convert the pyruvic acid into lactic acid. Lactic acid, sometimes known as lactate, just like pyruvic acid and pyruvate, kind of go interchangeably. Okay. Now, lactic acid is something you've probably heard of before. I talk a lot about lactic acid in my exercise physiology class. It gets a bad rap. A lot of people think it causes muscle soreness and cramps and that kind of stuff. They don't know what they're talking about. It does cause fatigue, though. Okay, so that is something that it is related to. Okay. What happens can be described down here with this figure. This is very similar to the figure that we saw for glycolysis. In fact, if I were to draw a line right here on the other side of pyruvic acid, everything to the left is the same thing we saw the other day. It's just a little bit different for glycolysis, where we go from the 6-carbon glucose, we add some ATPs to it to split it, we yield out a total of 4, but only a net of 2. There's our 2 ATP. We oxidized it to form some NADH plus H pluses. These NADH pluses, or NADH plus H pluses, are the key here. Okay, because from pyruvic acid here to here, this is fermentation. We take those NADHs that we just formed, and we flip it and do the exact opposite what we did during glycolysis. We end up giving those electrons and hydrogens right back to the pyruvates. That's what forms lactic acid. Okay, we can see that over here on the right. Okay, the hydrogens that are shown here in the kind of blue boxes end up there. What's not shown, but what happens, and you can see that here, are the electrons are also brought back to the pyruvic acid, and that's what forms lactic acid. Okay. Now, why is this done? Well, we're not forming any ATP here, are we? What we're doing is freeing up some NAD pluses. That way this vehicle can go back and find a new glucose to oxidize, and therefore make some ATPs through glycolysis. So fermentation doesn't directly make the ATP, it makes these, and therefore we can keep glycolysis going, which means we can allow new glucose molecules to go through glycolysis. Is this a very efficient and a preferred mechanism? No, it's kind of a last-ditch effort. Because we're only going to get a little bit of ATP, and as I mentioned, the lactic acid will build up. Okay. As it builds up, it'll diffuse out of the cell, enter our blood, where it'll make its way to our liver to hopefully be converted back to pyruvic acid. If it doesn't, it can interfere with things like muscle contractions. All right, so that's what happens if oxygen is scarce. If oxygen is plentiful, what we once again call aerobic conditions, we're going to convert that pyruvic acid to acetyl coenzyme A. Okay. Now, the textbook doesn't specifically call it the transition reaction, Some textbooks do. I like the phrase transition reaction or the term transition reaction because it kind of tells you what it is. This is a reaction that's basically between glycolysis and the Krebs cycle. It helps us get to that point. It transitions us. Because pyruvic acid cannot enter into the Krebs cycle, we must convert it to acetyl-CoA. And that's what this process is. What we do is shown here with the 3 steps at the bottom. We remove carbon dioxide from it. Okay. We're showing one little circle. Remember these gray dots or circles represented a singular carbon. Okay. And as you see, carbon dioxide, CO2, has 1 carbon. That means our 3-carbon molecule is now down to 2 carbons. Okay. Now, that carbon dioxide is important because, as we know, that's what we breathe out. And if you wondered where it's produced, here's where some of it is produced. We oxidize that acetic acid, which means that an NAD+ comes up, picks up some hydrogens and some electrons. And then the last thing we do is we slap a coenzyme A on it, forming acetyl-CoA. Okay. This takes place in the matrix of our mitochondria, which is also happens to be the same place that the Krebs cycle takes place. So for that reason, sometimes this kind of gets lumped in as part of the Krebs cycle, when in actuality it has to happen before the Krebs cycle can take place. The figure that I'm going to show you for the Krebs cycle will actually show this at the top. Okay. One thing that you have to remember is that this reaction actually takes place twice. Because how many pyruvic acids did we form from glycolysis? 2. What's only shown here is 1. So if I really wanted to be more correct, I would just put this figure on this page twice. That seems kind of like overkill. Okay. So as a result of this reaction, we formed 2 NADHs and 2 acetyl-CoAs. That way those 2 acetyl-CoAs can head for our next reaction, what we call the citric acid, PCA, or my preferred term, Krebs cycle. And this is going to occur in the same place. It's also aerobic, just like the transition reaction was. And this is a series of reactions where we basically take apart the acetyl-CoA, remove many of the carbons. There's where more carbon dioxide comes from. And it's basically just best shown in the image. Okay. Here's our textbook image. I actually pulled this one from our textbook. Okay. At the top here, right here, that I'm circling with my mouse, that's the transition reaction that we just talked about. Okay. This green circle is the Krebs cycle. And you can see why it's called a cycle. That's a very simplified image because they don't show what's actually happening. But right here where the cycle begins, there's a 4-carbon molecule that joins with our 2-carbon acetyl-CoA. Okay. And if you add 4 and 2, you get a total of 6. Okay. That's what citric acid is. It's a 6-carbon molecule. Okay. Now, the coenzyme A that was attached to the acetic acid is actually removed right away. It's just needed to get us in. Okay. So our 6-carbon citric acid runs. And you can see we lose 2 carbons right away through carbon dioxide. So if you lose 2 from 6, now you have 4. Okay, and that 4 is what will start us over again. That's why they call it a cycle. Okay. If you look over on the left, we say that there's 4 carbons formed, or 4 carbon dioxides formed. Okay, well, we only showed 2. We showed 2 ATP, but we only show 1. We show 6 NADH plus H pluses or NADH2s. We show only 1, 2, 3 over here, and 1 FADH2 shown, but we list 2 off. Why is our figure only half of what's listed over here? Because it happens twice. Remember, there were 2 pyruvic acids from our original glucose molecule. Remember that? Make sense? All right, let's watch a clip on that. Okay, so let's count them up. We're going to get a tally here of ATP first. How many net did we produce during glycolysis? 2. How many net were produced in the transition reaction? That's a trick question. The answer is 0. Okay. And then how many net were produced in the Krebs cycle? 2. So we had 2 and 2 together. That's a total of 4 through the first 3 of our 4 steps. 4 ATP is not very much, is it? Okay. What have we formed a lot of, though? These guys. Okay. Back in glycolysis, we formed 2. In the transition reaction, we formed 2. So that's 4. 6 more during the Krebs cycle, that's 10. And then FADH2 is like the cousin of NADH2. It does the same thing. And it's just a different vehicle that's going to carry those electrons. So we've got 12 total carriers of electrons and hydrogens. And guess where they're going to carry those electrons to? The electron transport chain. And this is where the magic actually happens. What the electron transport chain is basically can be best shown in the image here on the next page. It is a series of protein-carrying or electron-carrying proteins. Okay, and they make up a chain that carries electrons. Hence the name electron transport chain. Okay. Now there are these purple proteins that pass the electrons from those electron carriers. The NADHs and the FADH2s that we formed in our first 3 steps drop off the electrons here. They also drop off the hydrogens. Okay. As those electrons get passed down the chain, the hydrogens get pumped into this space here. Okay. This process takes place in the inner mitochondrial membrane, what we call the mitochondrial cristae. If you think back to the beginning of the semester, we learned that mitochondria had a unique structure. They had an outer membrane, which was really not that important to us. They had an inner membrane that was folded in on itself. That's called the cristae. Okay, and that's where this place takes place. Okay, inside of that, where the Krebs cycle and the transition reaction took place, was the fluid called the matrix. Now, between those 2 membranes is a very tiny little gap, what we call the intermembrane space, and that's what we show right here. So here's our interfolded cristae. Here's the outer membrane. That dark brown here is the intermembrane space. As those hydrogens get pulled across, we establish a gradient. And we know that means there's going to be a higher concentration of one thing on one side and a lower concentration of something on the other side. We have a hydrogen ion concentration, where it's higher here and lower here. And as we know, we always want to go down our concentration gradient towards equilibrium, right? Well, there just so happens to be, next to those electron transport proteins, an enzyme. This is the only enzyme in the entire lecture that you need to know, and that is ATP synthase. Guess what that enzyme does? Based on its name, it synthesizes or produces ATP. As those hydrogens are pulled through it, it energizes it enough to take ADP and PI and join them together to form ATP. And that's how it all happens. That's where the magic actually happens. Okay. So let's read through. Okay. Pairs of hydrogens that were once part of the glucose molecule are brought to these proteins by the carrier molecules NADH plus H plus and FADH2. Each hydrogen is then split into its proton, H plus, and its electron. Okay. We call hydrogen ions H pluses protons because that's basically all it is. Okay. If you remember back, we learned hydrogen had atomic number of 1, which meant it had 1 proton, 1 electron. Well, if its electrons pulled away from it, all it is a proton. So sometimes you'll see protons and hydrogen ions listed as the same thing. Okay. We pass the electrons from one protein to the next, Finally, oxygen is what accepts them. And remember, if you think back to the very beginning of the lecture, the first time we saw the formula for cell respiration, we said that oxygen was getting reduced. That ends up becoming the water that we talked about. Okay, the oxygen and hydrogen ions join together to form water. Okay, that is called metabolic water, okay, and it helps to contribute to our necessary intracellular fluid. It also can help prevent acidosis. Because if hydrogen ions accumulated, pH would drop. Okay. Now, the reactions of electrons with the proteins releases most of our energy that was once contained in the glucose molecule. So it has everything to do with the electrons that were in our glucose. Oxidative phosphorylation is the actual oxygen requiring joining of our inorganic phosphate to ADP to form ATP. Through the electron transport chain alone, this last step, we yield out 32 to 34 ATP. And that's a total from that singular glucose molecule. Now, I'm going to explain why in a little bit, why it's not 32, why it's not 33, why it's not 34. It's kind of an average somewhere in there. Now, the actual production of ATP in cell respiration takes place through a process called chemiosmosis. Okay? It involves that ATP synthase being moved down its concentration gradient from the intermembrane space to the matrix. Okay? Let's finish our video. All right, looking real quick, I'm going to bounce back to this figure. What you're going to see is that FADH2, bringing those electrons, doesn't actually bring out as much ATP as NADH plus H+, because it donates its electrons later on down the line. Therefore, only a few or less hydrogens get pulled into the space. Okay. But that doesn't affect the total number as far as why it's 32 to 34. Okay. You'll see that, and I'll explain it here in just a second. So let's read through the whole summary. For every molecule of NADH plus H+ dropping off hydrogen atoms to the electron transport chain, 2 or 3 molecules of ATP, which is an average of 2.5, are produced by oxidative phosphorylation. For every molecule of FADH2 dropping off its hydrogen atoms to the electron transport chain, only 1 or 2 molecules of ATP, which is an average of 1.5, are produced by oxidative phosphorylation. This difference is due to the fact that FADH2 drops off its hydrogen atoms at a lower energy level, complex 2, than NADH plus H+, which was complex 1, what I was just talking about on the slide or two ago. So, to summarize and to bring it all together, one glucose molecule will produce 36 to 38 ATP after completing aerobic respiration, okay, which involves the 3 major steps: glycolysis, Krebs cycle, electron transport chain. You know, I didn't list here, but transition reaction will be right here. Kind of not a super major stage, but it is a stage. Okay. We describe this whole process as aerobic respiration, which is the 3 processes just mentioned in the making of ATP with the use of oxygen. Summary of the 3 stages of cell respiration also requires vitamins and minerals that are essential for this process, hopefully helping to establish and bringing together the important relationship between eating and breathing. Eating provides us with a potential energy source, whether it's carbs, whether it's proteins, whether it's fats. And it's often glucose, as we've just been talking about, and with the necessary vitamins and minerals that help aid those processes. To release the energy from food, we must breathe. Oxygen we inhale is essential for the completion of cell respiration, and CO2 produced is exhaled. Here on the bottom left, I can explain why it's not necessarily 32, why it's not 34, why it's not 36, why it's not 38. It's kind of that or average. Okay. So let's just summarize and I'll explain it. All right, here in the top left of our figure is a kind of tan shaded region. This is the process of glycolysis, which as we see takes place out in the cytoplasm, okay, where we yield out a net of 2 ATP, we form 2 pyruvic acids or pyruvates, and we form NADH plus H pluses, 2 of those. Okay. Right here in this part of our figure, which is in the mitochondria, is our transition reaction. No ATP is formed. We did form some CO2, but we also, more importantly, formed NADHs. The acetyl-CoAs that are formed can then enter the Krebs cycle, where we form some more CO2. We form a lot, 6 NADH plus H pluses, 2 FADH2s, and a couple of ATPs. Okay. So, for those 3 steps, glycolysis, transition, and Krebs, together they form only 4 ATP. But they formed a lot of the NADHs, those electron and hydrogen ion carrying vehicles. Here's where the 32 or 34 number comes from. Okay. Those 2 NADHs that were formed in glycolysis have to be carried from the cytoplasm into the mitochondria. And sometimes that requires a little bit of energy. So if you see here, we list the amount of ATP coming from those 2 as 4 or 6. Whereas the NADHs from within the mitochondria, it's always 3 per. 2 times 3 is 6. 6 times 3 is 18. We only get an average of around 2 for the FADH2s. 2 times 2 is 4. So if you add that all up, 32 or 34, this is from the Krebs only, or the electron transport chain only. Together you add those, 32 plus 4 is 36. 4 plus 34 is 38. Now, are you going to need to know every single step from beginning to end? Yes, in theory. Are you going to have to know like all the steps involved with each of those, like the 10 steps of glycolysis that I mentioned? No. I do want you to know like the numbers of ATP, the number of NADHs, that kind of stuff, where they're coming from. So questions I might ask, how many ATP are produced from the Krebs cycle? How many are produced from the electron transport chain? How many are produced total? Okay, so if you study those inputs and outputs, that'll help you tremendously. Okay. If you take upper level science classes like molecular biology, you'll go through and learn all those 10 steps of glycolysis and the steps of the Krebs cycle, all that wonderful stuff that I'm not going to require you to know. So it's not nearly as hard as it could be. Table 25.1 over there on the right just basically summarizes what I just shown you in the figure and what's listed up there at the top. So that's carbohydrate metabolism. Okay. Here in a second, let's do anabolism. This is just the same figure basically that we saw earlier. All right, so catabolism once again was breakdown. Anabolism this time is build up. So let's read through. Even though most of glucose in the body is catabolized to generate ATP, meaning it's broken down, we can also use glucose, or it can take part in or be formed by way of several anabolic reactions. Because remember, catabolic, break down, anabolic, build up. If our glucose is not needed by our cells for ATP production, we can combine it with other glucose molecules to form glycogen. We learned about glycogen at the beginning of the semester. It's the polysaccharide form it's a polysaccharide form of glucose storage in our body. Basically, we take a bunch of glucose molecules and we bond them together, and then we store it as glycogen in 2 major places: our liver, hepatocytes, and our skeletal muscle. This is what we do, like when we talked about carbo loading earlier in the semester. That way, later on, if you need to, you can take that glycogen and break it down into glucose, which then can be used to make the ATP. The formation of glycogen is called glycogenesis. It's one of those 6 words I wrote up on the board and defined the other day. Literally has the word glycogen at the beginning of it and the suffix genesis at the end. Okay. Genesis means to form or build, so it literally is translating to formation of glycogen. Okay. This is what we do when we are hyperglycemic. which we know that term means having high blood sugar. And that is going to stimulate the release, as we know, insulin. Okay. And this is all review from what we talked about a month ago. Now, as I mentioned, when our body requires ATP, like during exercise, we can take that glycogen that's stored in our liver and in our skeletal muscle, and we can break it down into glucose, which then can be used to make ATP. And that process is called glycogenolysis, which also has the word glycogen in it, and lysis, which means to break it down. That is stimulated by glucagon and epinephrine. Glucagon, remember, I used the phrase to help you remember, we secrete glucagon when glucose is gone, aka hypoglycemia, low blood sugar. What's the kind of general term for epinephrine? Instead of calling it epinephrine, most people call it adrenaline, which we release during that fight or flight response, right? Okay. And during a fight or flight response, we need all hands on deck, aka we need as much energy as possible. How do we get energy? Well, we can break down our glycogen into the glucose, which then can be used to make the ATP. Okay. We can also make glucose from triglycerides and proteins in response to low liver glycogen and in a fasting state. And that's what we're showing down at the bottom. Okay. Glycerol, part of a triglyceride, lactic acid, and certain amino acids can be converted into glucose by our liver in a process called gluconeogenesis. Okay. So we're making glucose from non-carbohydrate sources. Okay. And that is stimulated by glucagon. It's also stimulated by cortisol. Cortisol was a adrenal cortex hormone that we nicknamed our stress hormone, which we will release if we are in like a low-carb diet situation. And then your thyroid hormones T3 and T4 actually will promote gluconeogenesis. Those three processes are all illustrated here on this page. Okay? They're a lot more complicated than I basically just described. But if you just follow the arrows, that's all I really care about. Okay. Going from blood glucose to glycogen with our red arrows, that is glycogenesis, okay, which is stimulated by insulin, okay, when we are high in blood sugar. The opposite, glycogen to glucose with our blue figure or blue arrows, when we are hypoglycemic or when we are exercising, glycogenolysis. Two major hormones involved with that, glucagon and epi. Okay. And then over here is gluconeogenesis, where we can take things like parts of the fats, lactic acid or amino acids, do some different things. Even pyruvic acid, we can go backwards and form glucose. Gluconeogenesis, formation of glucose from non-carb sources, primarily stimulated by cortisol and glucagon, with T3 and T4 aiding in that process. Okay. So you won't have to know the steps involved with any of that, but know what those things are and the hormones involved, which you guys should already know, because we already talked about this earlier in the semester. All right, that's it for carbs. Let's now talk about the other fuel sources. So what are those other fuel sources? Proteins and fats. So this is what we'll do if we don't have enough carbs in our diet, or if our carb sources are low, or if we are doing some other certain things that I'm not going to get into. Let's first talk about proteins as energy sources. We learned earlier in the semester that, and we've talked about this really recently, that proteins are made up of amino acids. Down at the bottom here I have a figure of a singular amino acid, okay, known as an amino acid because it has an amino group, and then the carboxyl group on the other side is the acid part. What we do to use amino acids as energy substrates is we remove this part. We remove the amino group, which is why we call it deamination. Okay. Deamination is where that NH2 is removed. You can see that right here That NH2 will then end up becoming ammonia, which is a very toxic compound for the body. Fortunately for us, and we've actually talked about this before, our liver will convert that to urea, which will then get filtered out and peed out, or even lost through sweat. Okay. So what's left over is the other stuff, which can sometimes be 2 carbons in length or 3 carbons in length. And then those can basically get plugged in right there somewhere close to the Krebs cycle. Okay, because keto acid and pyruvic acid, in this case, you see, and we'll talk about this a little bit more and you'll see in the figure, are very similar. Now, proteins are generally not a preferred energy substrate. Fats and carbs, though, we do like to use. Okay. So lipids, like carbohydrates, may be catalyzed or catabolized to produce ATP. If our body has no immediate need to use our lipids in this way, they're going to be stored as triglycerides in our adipose tissue throughout the body and our liver. So if we make fats, and that's one of these 6 words, we're performing lipogenesis. Unfortunately, typical American diet ends up with us doing too much of this. Okay. We can store a lot of energy in our fat, and that's why we do it. That way, if we require energy, we can pack a big punch by performing lipolysis, the breakdown of fats. Okay. And that's going to be through lipase enzymes, which break our triglyceride apart. Down at the bottom here is a figure we saw at the beginning of the semester, which is the figure of a triglyceride, made up of a 3-carbon glycerol. Okay, that's not as important as these guys. But this 3-carbon glycerol basically can be twisted around and converted to our pyruvic acid, which was also 3 carbons in length. And then from there it can enter into the Krebs cycle after it's gone through the transition reaction, and then head on down to the electron transport chain, and we can get some energy from it. More importantly is what we can do with these fatty acid tails. Each of these fatty acid tails can sometimes be 20 carbons in length. And what we do with them is perform a process called beta oxidation, where we take them and we split them into 2 carbon segments. Okay, so if you have 20 carbons in length, you can get 10 2-carbon segments from each of those. Okay. If you remember, after the transition reaction, we had a 2-carbon acetyl-CoA. Well, you attach a coenzyme A to each of these 2-carbon segments, you can throw it right into the Krebs cycle. Think about how many times you can run through the Krebs cycle from a singular triglyceride. Tons. In fact, reading through, since fatty acid chains can be somewhere around 20 carbons in length, 10 acetyl-CoA molecules can be formed in our Krebs cycle, providing enough energy to yield 100 ATP. Here's your answer to that question, Blair. We get a heck of a lot more ATP from fats than we can from carbohydrates. A singular gram of fat is actually going to contain twice as many calories, actually over twice as many calories as a single calorie or a single gram of carbohydrates. Okay. Calories, that word is a buzzword for people when they talk about diets. Most people, if you ask them to explain what a calorie is, they'd be completely clueless. Basically, just kind of simply, calories are a measure of how much energy you can get from a food source. And if you're eating fats, they're going to be really high in calories. To go along with fats and proteins are ketones. So reading through, as part of normal fatty acid metabolism, liver converts some acetyl-CoA molecules into substances known as ketone bodies, just ketones. This includes acetoacetic acid, beta-hydroxybutyric acid, and acetone. Formation of these 3 substances is called ketogenesis. Once they're produced, ketone bodies leave our liver, enter our body cells, where they are then broken down into acetyl-CoA, which then can be plugged right into the Krebs cycle. Level of ketone bodies in our blood is normally very low because of other tissues that use them for ATP production as fast as they are formed. But during periods of excess beta oxidation, production of ketone bodies will actually exceed their uptake and use by our body cells, which can occur after like a triglyceride-rich meal, during the fasting state, or starvation, because few carbs are available for catabolism. Excessive beta oxidation may also occur in poorly controlled or untreated diabetes. And if our ketone body levels go way up, it can cause a condition called ketosis. Now, those ketone bodies, most of which are acids, must be buffered. And if we can't buffer them, we're going to become acidic. And that's what ketoacidosis is. This decreased blood pH can in turn cause depression of our central nervous system, resulting in disorientation, coma, even death if the condition is not treated. This is actually what can happen if you go on a no-carb or low-carb diet, which is absolutely not a good diet to go on. It's going to cause you to lose weight, but it can really screw up all sorts of other things. What people look for is the easy route, and you can still get plenty of carbs. You just have to get the right ones. Fruits and vegetables with the carbs in those are good. So are your whole grains, your pastas, your potatoes, that kind of stuff, instead of the cookies and cakes that taste so sweet that are loaded with sugar. Now, the last thing here listed is related to diabetes. When a diabetic becomes severely insulin deficient, one of the telltale signs is a sweet smell on their breath from that ketone body acid. They even say sometimes that the urine will smell a little bit fruity, which is kind of crazy. All right, figures here on the last, this next page, not the last page, walks you through lipolysis and lipogenesis. Lipolysis is where we break down those triglycerides into their two components, glycerols and the fatty acids. And then it shows where they can be plugged in before the Krebs cycle. Same thing for amino acids. After we deaminate them, they can basically get plugged in right there at the transition reaction. Lipogenesis is the opposite direction, where we take those glycerols and fatty acids and we make triglycerides, stimulated by insulin, whereas lipolysis, the other hormones. I'm not too worried about the hormones for those. I didn't list them in the lecture. They're just here in the figure. And then over here on the right is kind of a very simplistic view of how we can end up with the electron transport chain, either from glucose, from amino acids, or from fatty acids, which are the parts of our triglycerides. So kind of a simpler view of this figure over here. All right, table 25.2 is a nice summary of the different aspects of metabolism. We spent the majority of our lecture talking about the breakdown of glucose, with the 4 stages: glycolysis, Krebs, transition, and electron transport chain. They didn't even list the transition reaction, but it's in there. Then we just briefly talked a little bit about anabolism of carbohydrates. We talked briefly about catabolism and anabolism of triglycerides, catabolism of proteins. What we did not talk about at all in this lecture was anabolism of proteins, because we've already talked about it. I didn't figure we'd need to do it again. Thinking back to the beginning of the semester when we learned about gene expression, where DNA can be used to make RNA, RNA then can be used to make proteins. That's protein anabolism. We've talked about that before, so didn't need to list it again. All right, the last bit here in our lecture is going to be about body temperature regulation, which sometimes is described as the term thermoregulation, something that's actually very, very tied into your metabolic rate or metabolism. So let's read through. Your body produces more or less heat depending on the rates of its metabolic reactions. Because homeostasis of body temperature can be maintained only if the rate of heat loss from the body equals the rate of heat production by its metabolism, or by metabolism. It's important to understand the ways in which heat can be lost, gained, or conserved. we talked about this at the beginning of the lecture. Okay. Whenever we perform catabolism, whenever we perform anabolism, heat is always given off. Great thing about us is we are warm-blooded. Bad thing about us is that we are homeothermic, which means we like a very, very constant body temperature. Okay. We talked about earlier in the semester, if temperature goes up or if it goes down, our proteins denature. And since proteins are a component of just about everything, our body will shut down if temperature goes up or down too far. Okay. Now, heat is a form of energy. It's actually a form of kinetic energy. You can't really see it. But we can measure it with temperature. And we'll talk more about core and shell temperatures here in just a second. Despite wide fluctuations in environmental temperatures, homeostatic mechanisms can maintain a normal range of internal body temperature. We have several different things that we do to help either promote heat loss or to promote heat gain. Because if body temperature drops, we need to increase the amount of heat we produce. And if body temperature goes up, we need to increase the amount of heat that we dissipate. And that's what allows us to maintain a constant core temperature near 37 degrees C or 98.6 degrees Fahrenheit. Since heat is a product of metabolism, it's important to understand metabolism. And we talked about that throughout the entirety of this lecture. Basal metabolic rate, or BMR, is a measurement of an individual's metabolic rate with the body in a quiet, resting, and fasting condition. And so on Wednesday, we're going to talk about ways that we can influence our metabolic rate and therefore influence our heat production. Okay. Let's talk just real quick here before I set you free about core temperature versus shell temperature. Pretty straightforward, common sense. Core temperature is temperature in our core. Okay, there's kind of some invasive ways to measure this, so it's not done as often. Okay, there's esophageal that's used a lot of times, where you actually run a line in through their nose and then down through their throat and their esophagus. It's actually better than the other, which is the rectal core temperature. We have to go up instead of down. Okay. There's an even easier and kind of newer method where you can actually have people swallow sensors, kind of like a pill, and then they end up defecating them out afterwards. But they are all wireless, and so that's pretty nice. But when you go to the doctor, they're not going to stick anything up or down or have you swallow anything like that, because they make those very easy to use. Shell temperature thermometers, like the ones that measure your temperature on your forehead or the side of your head. What they're doing in that case is averaging, hopefully, or finding your shell temperature and hoping it's somewhat close to your core temperature. Fortunately, shell temperature is not always very indicative of your core temperature, because it can vary very easily because of the outside environment, how that can influence it. Okay, especially if you come in from like an outside that's really cold, make your core temperature much lower than your core temperature. Your shell temperature much lower than your core temperature. What we're going to talk about, like I said, on Wednesday is how we can increase metabolism to help match heat production with heat loss, or to help dissipate heat. Okay, several factors influence our metabolic rate, therefore rate of heat production. Exercise obviously is going to make your metabolism go up, heat go up. Same thing for thyroid hormones, epi and norepi, when we have the sympathetic nervous system activated. Your body temperature, ingestion of food, age, gender, climate, sleep, and malnutrition can all influence metabolic rate and therefore. All right, we'll call our day here. Pick up more with temperature regulation on Wednesday.